Hello, my name is Jansen. I'm a postdoc at the University of Tasmania in Australia, and I'm going to present to you the work that we're currently doing on creating a circumpolar database of high-quality biological and environmental data that we want to use to map the distribution of Antarctic seafloor biodiversity. The motivation behind this project is that the fauna on the Antarctic seafloor is unique and highly biodiverse, but their distribution is still not very well understood. And this is a problem because not knowing where these animals live hinders effective conservation and management. I've added a few nice pictures here just to highlight how stunning these communities really are. Okay, next slide. So while we know very little about the distribution of the biodiversity, we do actually have all the tools available to make a big step forward. And that was the motivation for our project, to map the distribution of the Antarctic seafloor biodiversity on the entire continental shelf for the first time. And there are three main efforts within this project. First of all, gathering all the biological data from images. Second, generating a database of environmental variables. And then third, analyzing these data together to predict the distribution of species, biodiversity, vulnerable marine ecosystems, and also estimating the impact of seafloor fauna on carbon capture. My focus today in my talk is on these first two work packages, while Nicole Hill has a talk that focuses more on the application, so everything on the right side here. Also, Victor Shelamov, he has a fantastic talk where he goes into details about scoring the images and building the biological database. Okay, so first of all, I'll give you a quick overview of the biological data. The still images in this database are provided by scientists and collaborators from around the world. We have a total of about 60,000 images, of which we selected 3,600 for annotation. That equals to about an image every 100 meters of transect. Now we use two main methods to annotate these images. First of all, we overlay a point grid onto the images and identify what's located under each point. That gives us estimates of percentage cover of both animals and substrate. And the second method is to identify the location and circle each small animal because they often get missed by the point grid method. So in total, we have about 130,000 annotations of mobile animals and 250,000 annotations of percentage cover. Now, what's really important and what makes this database really unique and valuable is that each individual annotation is reproducible and can be easily edited. And that means if someone, for example, wanted to look at all the pencil urchins that we've annotated, they can just look at a folder which has all the annotations, has all the references to the images where these annotations were made, and if they wanted to, or if they have the expertise, they could then identify these pencil urchins into species or whichever group they want to. I can only recommend looking at Victor Shalamov's talk, in which he details a lot of the annotation and building the database for the biological data. Now, the second part of this project is about the environmental data. And today we already have a range of fantastic environmental data available, such as depth, slope, topographic position, etc. And also global estimates of net primary production that work well in the Southern Ocean. However, we're still missing important variables at a high resolution, such as seafloor current speeds and tidal movements, temperature, and we also don't know about the availability of surface-derived food at the seafloor. And this is really the key bit in this project, is that we've shown the proof of concept of how to estimate the availability of food at the seafloor by using satellite-derived surface production, ocean currents, and particle tracking, and combining this with diatom abundances from sediment grabs. And we are now combining this proof of concept We have a high-resolution ocean model that is being developed by our collaborators, and it's 2-kilometer resolution, has tides and everything in it. And this is really a fantastic step forward to understand the distribution of biodiversity on the Antarctic continental shelf. For the validation, we use diatom data that's being compiled and analyzed by Amy Leventer, and that comes from 21 cruises, also dating back to quite a long time ago. This is a fantastic resource, and we're currently still working on the food availability model, but the key outputs that we expect to have from that, and that'll be available to everyone sometime next year, is we will have the flux of food along the seafloor. We will be able to map sedimentation patterns, identify sources and sinks of these food particles, and also we will be able to map connectivity between regions, so using the ocean currents to identify which regions are connected to other regions. We're still a bit away from presenting any distribution maps, but what I can present today are some preliminary results from an analysis looking at which environmental space our data covers. And what this analysis does, we're using depth, slope, seafloor current, speeds, and net parameter reduction, and the sample size of the downward-facing still images to look at what the environment is that the samples were collected in, and what other environmental conditions are on the continental shelf. So in blue on this map and below, you will see all the environmental cells that have similar environmental conditions to the sample sites, and in orange are all those cells which are different. What we noticed is generally there's a good spatial coverage. There are some gaps noticeable on the eastern Arctic continental shelf where we have fewer data, and deep sites are disproportionately missing. Also, we have high current environments that are underrepresented, and you see these in these bar plots on the bottom left. We have the sample sites, and the environmental cells that are similar to sample sites and dissimilar in blue and orange for depth, slope, net parameter reduction, and seafloor current speeds. Now in the future analysis, we'll include seafloor food availability and use high resolution environmental data to get the actual estimates of how much of the seafloor environment on the Antarctic continental shelf we actually have covered with the samples, and then we can highlight which areas might be really important to sample in the future. So to summarize and give a bit of an outlook, the database of annotated seafloor imagery, we're still working on that, but it's going to be a fantastic resource available sometime next year. It's going to be fully reproducible and openly accessible for everyone. To go along with that, we have the environmental database at 2-kilometer resolution that's nearing completion as well. It's also going to be available to everyone, and so these two combined should be a fantastic resource to really make a big leap forward in understanding the distribution of Antarctic seafloor species and biodiversity. We also use the gap analysis to identify areas that could be priorities for future sampling efforts or monitoring. And all of this is really only possible because of the fantastic collaboration that exists between all the Antarctic scientists. So big thank you to everyone that's involved in this project, and we welcome anyone that has information or wants to contribute or wants to look at their pet species to please contact us. And please share your images, please share your data. We commit to do the same, and it would have been fantastic to foster that even further. And there's so many new studies and big picture questions that we can ask once we all share our data. A big thanks to all the collaborators, students and supporters involved in the project. Amy Leventer, Dave Bowden, Julian Good, Dieter Piepenburg, Craig Smith, Alex Post, Thomas Windsor, Fanny Hamann, Charlie, and probably someone that I forgot, I'm not sure. And don't forget to check out the presentations by Nicole Hill and Viktor Shelamov. Thanks for listening in, and have a good time at the online conference.